if we go ahead and take it in class, how long will it take before you get it from Blackboard?
How long? What now?
Grading?
Oh, for my grading? Oh, it'll be pretty quick. If whether you do, I mean, Blackboard's going to grade it instantly. If you took it online, all right. I'm going to have it graded within, by Thursday, at the latest. It's my guess. So, all right. Are we good? Now, I had cut this, and I just had to change the coloration because I had such a bugger all hard time getting the video to pick up the screen. So, and I picked it up this morning because I actually hadn't taken this home, hadn't anticipated this problem, and now, and I couldn't get my slides loaded to swivel, so there won't be any split screens. I'll either put this I'll probably do both. I'll probably put this video on swivel and I'll put it on YouTube after I get converted, if anybody needs it. All right, but that should be I should be able to get that done today. So, all right. So, chapter 24 is again one of those, hey, why don't we do this too, right? Why don't we talk about genetics now that we've finished with all these, because you guys clearly haven't had enough things that you explored in this semester, and we just got to throw this in too, because it makes three chapters on a test. Anyway, all right, so whatever the philosophy is, right, here's our reality, right? We've got to understand now that we've gone through this reproductive process and this development of our young in chapter 23 and their birth. We want to understand about how they survive and what traits do they have. Okay, so and for all the things that I'm going to write and have written on your test, because I've written the multiple choice part, a lot of this is definition. All right, you should know what a genome is and what a gene is, and you've got a review sheet for this too that's up on Blackboard, or has been up on Blackboard, and it's pretty straightforward, this chapter's questions. All right, so I do remind people that in every cell, right, because you all started as that zygote, which was the result of dad's 23 chromosomes and mom's 23 chromosomes. So all of your cells that you currently have came from that one cell. So they all reflect those 46 chromosomes. You learned in AP1, they're housed in, right, the nucleus. That's where DNA is kept because it is the chemical instruction sheet. It carries in there, right, so we have, as humans, and there are a couple numbers I think on the test this time around. As humans, we have 3 billion base pairs like the adenines and thymines and guanines and cytosines, right? We got 3 billion of these, which means your DNA is actually about 2 meters long if we took it out of your nucleus, right? With your nucleus being only 5 microns in size, okay? And in that 3 billion base pairs, there's about 20,500 genes. A gene is a piece of the DNA, roughly between 10 and 15,000 bases. And a gene is basically nucleotide information that codes for a protein. That's the simplest definition I can give you. The definition is evolving as molecular biology evolves, but that's the most straightforward way I can describe a gene to you. All right? So this just reinforces, again, what I've talked about. Now, genes can have different forms. That's where we use the term alleles. When I say, so there's an allele for this trait, hair color, eye color, skin color, height, right, heart disease, right, cancer. When I say there's allele, that means that there are variants, different forms of that gene, right? Because the gene is, right, it's a chemical code. There's different forms of it. In other words, there's different sequence for it, right? We also refer to this as polymorphism. Different forms, polymorphism, many forms. So in this chapter and in lab, if you had an opportunity to do this lab, we didn't on Wednesday. So the lab was reinforcing the idea of Mendel's inheritance, which was you have 2 alleles for a trait, just 2, and one of them is dominant and one of them is recessive. And I will address this in today's lecture. Most of the time, most of your traits are not simple Mendelian genetics. Most of them are not. But because this is an introduction to genetics, that's what we talk about. And so we refer to these as, again, the alleles. Mom gave you one, and dad gave you another. Okay, so you'd sit there and say, okay, there's only 2 then, right? Because you only have one mom and one dad. But remember, though, that we can have sets of alleles for a trait. So when we talk about something like skin color and height, right, we can have 3 or more sets of alleles. And a set means one from mom and one from dad, set or a pair. And how do I know this when I look out in the public? Because that's how we studied genetics. Genetics was a field that began well before we knew DNA sequencing, right? We just figured out DNA structure in the 50s. We only figured out genes after that. And we continued to figure it out. But geneticists, Mendel was in the 1860s, and Thomas Hunt Morgan was in the 1920s. So classic genetics is done just by looking, observing the population of an organism. And here we're talking about people, because this is human, AP. And so when we talk about this inheritance patterns, as we will later on in this, what I want you to understand is, as many of these inheritance patterns are not just simple Mendelian genetics. Right? They're often several sets of alleles that all influence each other and ultimately tell you what the trait is going to be. And the trait is always your appearance. What does it look like? As I say, traits or phenotypes are what do the genes look like on you. Okay? Now, how would I know this by observation? It's very straightforward. If I had something, a trait that was just simple Mendelian genetics, like attached and unattached earlobes, widow's peak or straight hairline, freckles or no freckles, bent little fingers, straight little fingers, hitchhiker's thumbs, interlacing fingers, dimples or no dimples. I know those are simple Mendelian because there's only 2 traits in the whole population, isn't there? There's only 2. You either have freckles or you don't. Right? You either have unattached earlobes or you have attached. There's only 2. That means there can only be 2 alleles. Right? If I talk about height, well, we don't have just short people and tall people, do we? So I automatically know that that's not simple Mendelian genetics, don't I? I know something else has to be going on because instead I have these people that are all over the place. Same thing for skin color, eye color, hair color, you name it. Most of your traits are not simple Mendelian. But when we teach it, in order for us to give you an introduction to it, we start with Mendel. Because he's still the father of genetics. And he still figured this out laboriously with thousands of people in it. We've just added to it. Okay? But genetics is fundamentally science and scientific method, just observation. And then trying to figure out why. So why did this show up? All right? So DNA, of course, as you learned in AP1, it's those nucleotides, right? These bases, carbons, hydrogens, oxygens, nitrogens, attached to phosphates and sugars, right? RNA is just a different sugar. Genetics, of course, is how do I pass this on? I want to understand, and that's why I talk about patterns of inheritance. And again, I couldn't care less about what color your children's hair will be, or how tall or short they will be. I do care about whether or not they have neurological diseases, or heart disease, or cancer. And that's really where we put our attention in genetics. So for all the super fabled information that you hear about genetics, genetics is really a field interested in understanding how we pass on disease and problems. All right? Genomics then is looking at, right, all these genes and how they influence each other. That's where genetics becomes really complicated. Because I'm going to tell you things like height and skin color, not only have we have more than one set, right, of alleles, mom and dad is a set, more than one set that's inherited, we also know that the environment affects it. And that's true for a lot of disease, right? Disease, ultimately, I say I can trace it back to your genetics, but I can't tell you, even if you have the correct gene or the incorrect gene from whatever viewpoint you take, that you're going to get the disease. The disease is a trait. I can't tell you that because there's a complexity of how do genes interact with other genes, which are inherited as well, and how does the environment influence all this. All right. This is just a nice picture to remind you that, yeah, right, we've got to package all this. We package it in these things called chromosomes. In chapter 22, we talked about this process, meiosis. Meiosis, of course, was I replicated the chromosomes. They're held together by a centromere. This happens whether it's meiosis or mitosis, right? Both of these processes do this, all right? So we've talked a little bit about DNA, that we've done it in the packaging that we call chromosomes in this class. In AP1, you should have had a full chapter, chapter 4, that at least half that chapter, that talked about the process of replicating DNA, right? Gene transcription, gene translation. All right? DNA, we can get 2 meters of this physical molecule into a nucleus that's 5 microns because we wrap the heck out of it. All right? It's wound around these proteins called histones, and it's very, very orderly packed. Because if we did this like our Christmas lights, right, and some of you might be stringing those over the weekend and you put them away, you might just crumple them up in your frustration and throw them in the closet, and then you have to deal with it next year, right? But you can't do that with DNA, right? Because DNA contains the chemical information for a gene. So I have to be able to go back and access that information. So this winding and packaging is a very precise process that's done in the nucleus. And again, nucleus of every single cell in your body. All right, so here's a consequence when we talk about genes. So here we've got chromosome 7. Now, of course, this individual would have 2 copies, right? Because we have 2 of each chromosome. We have 23 chromosomes in total, right? 23 pairs, 46 in total. Be careful with my language here. So here's just showing you 1 copy of chromosome 7 in this individual. And they use 7 here because that's where the gene for cystic fibrosis is found. Genes are found at specific locations. We call it chromosome mapping. We refer to their specific locations as a locus. Not the locust, but the locus, all right? They have a physical address. Long arm, short arm, what's the chromosome number they're on? That's what chromosome mapping does for us. Now, this shows you this process. Why is this gene so important? Well, because this gene becomes, right, here's it as DNA. It becomes transcribed into an mRNA. mRNA becomes a protein. This is a classic protein. In this case, the protein is a channel. It's a chloride channel in the membrane of cells. Now, you guys with a normal, or what we also call a wild type, normal gene for chloride channels, your protein that's made folds correctly, and therefore it transports chlorides, right? These negative charged anions, chlorine. And we know now that chlorine pretty much follows sodium anywhere in the body. Not in people with cystic fibrosis. Because if you have cystic fibrosis, your genetic information is incorrect. So the protein you make doesn't fold into a channel that allows chloride to move. It folds and it's stuck in the membrane, but it won't move chlorides. And because it doesn't move chloride, it changes the viscosity, right? Because remember those ions carry water around with them. Changes the viscosity of mucus and secretions, especially in the respiratory system and the pancreas. Digestive and respiratory are the #1 targets for people with cystic fibrosis. So, just a mistake, right, in the gene sequence leads to a mistake in the protein, which means that they don't function.
That's irreversible, right?
That is correct.
Anything
Now,
like that
we do gene therapy on people with cystic fibrosis.
after the fact.
Yeah, after the fact. So we
Like
can't.
you can't go into the fetus and
No,
do anything
this-
to reverse this.
It would be highly, highly experimental and questionably ethically.
Right.
Because I talk to anybody when you do a gene therapy, that because we said that all these genes have interactions with each other, and a lot of them we don't know even yet.
Kind of like mutations.
Yeah, we might be influencing something else. So we have to be very careful. And because our gene therapy is so crude at this point, I honestly refer to it as shotgun therapy, knowing what I know about it from doing research. Right? Is that you push in this normal copy, right, in here, and it might integrate in the right place and it may not. We don't precisely go in there, right, and stick it right in the right location. We pretty much overwhelm the chromosomes with a lot of copies of the normal gene and hope that it works. Right? Because the way we deliver this, of course, is through an inhaler.
Right.
And we put it in a vector like a virus. But a non-viral virus, if you will, or a non-virulent virus, that's pretty much adenoviruses are used, baculoviruses are used. Okay? It's not a perfect science, but it does work. All right? Because all you have to do is produce enough normal copies for them to retain function.
What'd you call it?
Gene therapy?
No, what was that last term?
Normal.
Normal function? No function?
Is it baclofen?
No, baculovirus.
Oh, back.
Yeah, vectors. Vectors.
Wait a minute,
Mhm.
what?
Yep. Vectors. Okay? So it's, right, this is what we face though, is the desire, once we understand what a gene is doing, and especially an abnormal gene, yeah, we want to figure out the trade-offs. And so this is one of the cystic fibrosis we do gene therapy with.
What about in vitro? Do they test the embryos or anything like that?
I am not aware that we've gotten to that point again because a lot of our sequencing is still questionable for privacy issues. What do we do with the information?
Yeah.
So it becomes very challenging. Yeah, you could. I mean, because as we'll talk about with the fetal sampling here at the end, amniocentesis and chorionic villi sampling, you can get the cells. That's how we figure out chromosomal abnormalities. The problem is then what do you do? Okay, so it's not that we couldn't do it, it's just there'd be, of course, a cost and there'd be privacy issues then. And then once we figure it out, what are we facing? And can we afford that? Can we not afford that? I mean, there's a lot of, and what, you know, again, rights come into the individual rights and so forth. But anyway, lots of very provocative questions for us. Much easier to do when you work with animal collies, let me tell you all right? Much easier, right? So modes of inheritance, this is what I was mentioning about those patterns. How do we pass these on, all right? So I say again, just to remind you again, meiosis, this is why chapter 24 comes here. Following meiosis, which was this, right, we did this crossing over, we did this, some independent assortment of chromosomes. We created an opportunity for a lot of genetic variation in meiosis. And that's good for our species, all right? So here, again, when we go back to meiosis and we say, okay, these combinations that happened, recombinations that occurred in mom and occurred in dad, and then mom and dad, two separate individuals with their own genetic components, though all human, right, get together, have a child, right? So joint fertilization, there's our zygote. So we can figure out the probability, the likeliness, and that's what we're really talking about when we talk about genetic counseling. What is the likeliness that a certain trait will appear between two different individuals, mom and dad? All right? These are the modes of inheritance, the patterns, because it's all pattern identification that we're interested in. All right? So this is a karyotype. If you've never seen one before, this is an actual photograph of the banding appears because they've colored this for mapping, chromosomal mapping, to show that these are actually homologs. So here we were looking at chromosome 7. I said we only had one copy there, so there is a second copy. So we refer to these, this is a female, got locked up here, but these are two X chromosomes. So this is a female, yeah, said down there, female, two X chromosomes. 23rd pair is the sex chromosomes we'll talk about. And so she had a maternal and a paternal chromosome, right? So when she was formed at fertilization, the sperm brought 23 of these, and the egg had 23 of these. So when I look at an individual, any one of us in this room, or this woman here, we are the sum of the maternal and paternal chromosomes. And it's 50-50, isn't it? All right? So I say that terminology-wise, because there's going to be a little bit of that on the test, pairs 1 through 22 are called the autosomes. So from here, row, they get lined up, put together. Here, autosomes. And they're homologs, right? Because each maternal and paternal look similar. They're similar in their size. They have a similar banding pattern. They carry the same types of genes. So if mom gave this young man back here, whoops, sorry, mom gave him a gene with cystic fibrosis, dad would have the same gene in the same position. Okay, so we say when we talk about these pairs of chromosomes, they're homologs. Right? So same types of genes as shown by the banding pattern. Centromeres end up in the same place and they're about the same length, clearly because, again, they have to have the same numbers of genes. All right? And this is where those 20,500 are divvied up. So each large chromosome especially has more genes. They top out, you know, 1,500 or so genes, maybe up to 2,000. Right? Smaller ones have less. Yeah.
So
You're physically looking at the amount of information when you look at a smaller versus a larger.
what numbers here are going to be important? You said there are going to be numbers on this.
Well, like the 20,500, the 3 billion, giving you the perspective of human. So I've already mentioned those, right? Knowing that chromosomes 1 through 22 are autosomes. Knowing the 23rd is a sex chromosome. And they're called sex chromosomes because when we originally discovered them, we thought the genes they carried only conveyed whether you were male or female. We didn't think there was anything else on them. That there weren't any other genes, just genes that said, oh, grow breasts or grow a penis or do whatever. That is relevant to sex or gender. Okay? That's why they got their name. These are called autosomes, again, because they carry these genes for all the other parts of our body on them. Now, the reality, though, is all 23 have to be found in all the cells of your body, right? Except for the cells that are actually in meiosis. Because they're, right, ejecting, not rejecting, but they're ejecting half the material by the time we get through meiosis. Okay? Is that, again, it's real important to have a visual when we talk about these things. All right. So, oh, that's a little too dark, don't it? I was looking at my office and it was, okay. Should have done it yellow. All right, so a gene exists, right, in these variant forms I mentioned, alleles, because we don't have just one sequence for hair color, eye color, attach or unattach earlobes even. It's not just one sequence, right? There's a variant in there. Somewhere in there, there's a difference in those ATCs and Gs. Their order, okay? So, here's our reality. We use the terms homo and heterozygous. I promised you zygous because that's the zygote. That's where we all began. So what does this actually mean? It means that when those 23 chromosomes from mom and those 23 chromosomes from dad got together in you, the zygote, were mom and dad giving you the same allele? Or were they giving you different alleles for this given trait? Because they can, right? There's only so many alleles we have to be humans, right? I don't have alleles for purple or yellow or orange skin color, do I? You see what I'm saying? So when we teach simple genetics, we want to first and foremost start with, did mom and dad give you the same allele for this trait, or did they give you a different allele for the trait? Homo, same. Hetero, different. Right? Simple definition. All right? Okay. Now clearly, mom and dad are not going to have the same alleles for all the traits, right? Because only identical twins should hypothetically have that. And identical twins are the same sex, so you can't have mother and dad that are identical twins, right? But with so many traits, some of them are the same. Right? Some of them are homo and some of them are hetero. But that's what it's trying to, again, getting back and imagining this idea. Going back here and go, okay, if this is the same, if it was the very same, well, that would be, you'd be homozygous for that trait. And if there was only one set of alleles. If they were different, mom gave it one variant, dad gave the other, you're a heterozygote. Okay, and we're going to have to go through and do this for each trait. So we can't just call you a homozygote, right? In general, we have to say a homozygous for this trait, or heterozygote for this trait. All right. The combination of these gene variants that we call alleles is your genotype. Right? So genotype genes. What are the actual sequences? Now, here's the reality. I told you that genes are about 10 to 15,000 nucleotides. So instead of writing out 10 to 15,000 nucleotides, which would take me from now until at least the end of this, right, and our time together, I'm just going to use a letter. Right? I'm just going to use a letter to represent it. It's just a symbol. And I can use any letter I want to or any symbol I want to. You see how this works? I mean, it's anything you want. Okay? But I'm not going to write out 10 to 15,000 bases because that would be kind of defeating the whole purpose of studying this, right? All right? Now, these genes tell me, or are going to tell my body, what protein to make, aren't they? These genes are going to be transcribed into mRNA, and mRNA is going to be translated into a protein. And a protein is my trait, right? The color of my hair and eyes and skin is all based on melanin. It's a protein. But that protein came from a gene. So when I talk about the trait or the protein, I want to know about your phenotype. And you can also, if that you don't like the P for P, it's also physically. What do you look like? As I say, off the, tongue in cheek, what do the genes look like on you? That's your phenotype. So when I say attach or unattach earlobes, that's a phenotype. But there's specific alleles that led to this phenotype, and that would be my genotype. So I want to be very careful, again, in just knowing vocabulary. Now, often, like in the cystic fibrosis, we use wild type and mutant, because the cystic fibrosis was a mutated, it was a changed, right, gene sequence. Wild type is just something preserved for the most common and usually normal gene variant. and mutant just means a change. I'm not going to ask a test question to get you to differentiate these two things, all right? I bring it up because when you look at some of our pedigrees here in a second, they'll use that wild type and mutant, and I'll explain that again. Now, so we get building on our vocabulary, homozygous, heterozygous, phenotype, genotype, and alleles. Now, when these alleles get together, right, you, the zygote, when they get together, how do they influence each other? Now, of course, if they're the same allele, the influence is not going to be a big deal. But if they're different alleles, then we want to figure out, well, how does that influence the trait? How does this influence what it looks like on you and how you act and behave and what diseases you have? So here's where we coin this idea of dominance and recessiveness. So dominant alleles, right, remember that's a symbol for the genes, there's a variant in the gene. Dominant alleles mean that they prevent, right, they dominate the phenotype. It doesn't mean they're the most common in the population, it means they dominate the phenotype. That their influence when they're together with another allele is that they mask its transcription and translation. And therefore, I never know you have this other allele that I'm now going to call recessive. Because I can't see it. Right? It's recessive. It's receding from me. So dominance and recessiveness refers to the interaction of these two things together. All right? So I say here again, to caveat this, is in many genes, the heterozygote, which means I have different alleles for this trait, hetero, different, one will determine the phenotype. And this was how Mendel's work proceeded. Now, it gets more complicated than this, but we want to start off simple, and it'll be good. So dominant prevents the recessive alleles phenotype from being realized. So the reality is I only see a recessive trait, like attached earlobes, or straight hairline, or no freckles. I only see those traits if the individual is homozygous recessive. I can't see them any other way, right? Because unless I have two copies of the recessive allele, I'll never see that trait. That's what Mendel discovered for genetics, and Dr. Morgan reinforced. Right? And that's what I'm saying here. Recessive allele is only expressed if it's a double dose, in the simple scenario we're looking at. A double dose. Homozygous, in other words. So, now I want to look at autosomal, sex-linked conditions. And all this means is where I'm finding the alleles. That's all it means. When I say autosomal recessive, autosomal dominant, patterns of inheritance, I'm saying simply that here's how the two interact when they're together, and they're found on chromosomes 1 through 22, because those are the autosomes. If I say your inheritance pattern is sex-linked, then I know that the alleles are found on either the X's or the Y's. Okay, so all I'm adding here is a location, a chromosome location. And I'm not very precise either, by the way, in this location. All right? And I say, how can I easily identify these in a population? Well, here's how it works. Because Thomas, again, in the 1920s, with fruit flies was his model organism. Thomas Hunt Morgan began just looking at breeding these flies, and he reinforced a lot of what Mendel had found. But then he started realizing, and he did this with eye color first, that this recessive trait that led to white eyes, right? So eye color trait. And we had these, right, phenotypes, red and white eyes. And white eyes was abnormal and wasn't seen, right, because it was recessive. It wasn't the normal. So dominant normal, dominant recessiveness. White eyes just wasn't seen unless you were homozygous recessive, so they're not as common, right? But then he also realized something else. He realized that the white eyes showed up mostly in males. And he couldn't make sense out of that, or the lab couldn't make sense out of that. Why males? I mean, what about that? And that's where, again, as they investigated further, they realized that, the allele is actually found on the X chromosome. So the allele for eye color wasn't related to gender, but was there on the chromosome that we thought was related to gender, right? So why is this a big deal? Well, males only get one X. So a male can't really be a homozygous or a heterozygous, by the way. It's only one copy. He either gets a normal or he gets the abnormal. Okay, and that's the foundation to sex-linked traits that we're going to look at here. So I say, how did you ID these in the population? You just look around. So what are some of our X-linked traits? Well, color blindness, hemophilia, Duchenne's muscular dystrophy. They predominantly all show up in males. Not exclusively, you can get females. It's just that a female, again, it's a recessive allele. So a female has to have 2 copies. And it's just less common because you have to have 2. Male just has to have 1. Yeah, it's just math. All right, so this is looking at the Punnett squares and pedigrees, which are the tools to figure out inheritance. That's what they are. They're just tools. Here's the wild type and cystic fibrosis. So we're looking again at a couple here. All right, now, here's their kids. It doesn't say they have 4 kids. We're looking at probability, likeliness. All right, so mom and dad are actually heterozygotes. So it's a little bit light on that background, but they're wild. They have a wild type allele, and they have a mutant allele. That's why I said we can use this symbols too. Letters, uppercase, lowercase, pluses, minus. I mean, it doesn't matter, right? Because they're just symbolizing a gene that is 10 to 15,000 bases, right? So here, mom and dad are heterozygotes, and this is autosomal. I'll let you know that right now. This is not carried on the sex chromosomes. So, and we call them carriers because the recessive allele is present, but it doesn't contribute to the phenotype, right? Because we have dominant. They're heterozygotes, and the dominant one masks, prevents the recessive one from being seen, being realized. They have children, right? And this is just probability. So I either talk in ratios or I talk in percentages when I teach this to students. It's just probability. So a Punnett square lets me organize the probability. So what do I do in a Punnett square? Well, first and foremost, I know when mom and dad got ready to have sex and have these kids, they had to go through meiosis, which meant they were going to separate those 2 homologs, weren't they? They have a set of alleles, but they can only give 1 to each child because they only give 1 chromosome. Remember that? That's meiosis. Only 1 gets to go. And it is genetic lottery which one goes. Right? But they're going to separate. So a Punnett square down here is just a way of organizing the alleles that's separated in meiosis. That's all it is. It's just a tool. So here I say, okay, in mom's eggs, oocytes, dad's spermatocytes, this line is the line between them. Half of the oocytes in mom would have the wild type or normal gene. Half of them would have the abnormal. Same thing for dad, right? Half of his sperm would have normal or wild type allele. Half of it would have the cystic fibrosis allele. And I could substitute any symbol I wanted in here, couldn't I? Because the Punnett square is just there to organize the alleles, period. And then I just do, hypothetically, what would happen in their kids. And I say, okay, mom and this, if this is the zygote, because each of these squares is a zygote, isn't it? If this oocyte was fertilized by this sperm, I'd have a child who was a homozygous wild type. If this sperm was fertilized by, or this sperm fertilized this oocyte, I'd have a heterozygote child, but still normal, if I want to look at the phenotypes and genotypes. All right, if this sperm was fertilized this normal, right, this mutant fertilized that normal, child's a heterozygote. If this sperm fertilizes this oocyte, the child is a homozygous recessive and has cystic fibrosis. That's all a Punnett square does for me. Is I'm organizing the alleles, and I'm only ever going to do for 1 trait with 2 possible alleles for you guys. All right, 1 trait and just 2 choices, so 4 squares. Yeah, Mark.
You're saying wild type, you just mean normal, right?
Yeah.
Okay.
That's just the term for it. Yep. All right. So now, what does this tell me? It tells me, right, if I want to know about the genotypes of the kids, the possible, because this is a possibility. This isn't reality, is it? Until they start having kids. So, in this situation, if I want to know the genotypes, I would say that, right, 1 in 4, or 25%, 1 in 4 is homozygous dominant. Okay? 2 in 4, or 50%, are heterozygotes. And 1 in 4, or 25% of the kids, have cystic fibrosis, or let's stick with the genotypes, are homozygous recessive. Right? if I wanted to know the phenotype is do you have the disease or not, I would say that 1, 2, 3 out of 4, or 75% are normal, and 1 in 4, 25% have cystic fibrosis. So I've just given you how we report the findings from simple genetics. I want to know what are the alleles if I ask for a genotype. If I ask for a phenotype, I want to know about the trait. And it's going to be something based out of 4, right? You know, I mean, and that's what it says, students. You got a chance of 0, 25%, 50%, 75%, or 100%. And there is nothing else in between there, is there? There's no other way to divide up those 4 things, is it? Right? There should be no crazy answers like 33 1/3%. All right? None of those. 80%. No. I'm just looking at probability here. Now, this is a pedigree, and I won't make you guys do these. I'll make my general biology students do these, but this is really what a genetic counselor does, because a Punnett square is just for this couple. A pedigree is taking it through generations. So in this case, it's parent to child, but in a pedigree, we could add grandparents and great-grandparents. If we had the information, we could make this pedigree huge, right? And that's why there's a lot of services out there that'll sell you, right, the software to be able to generate pedigrees based on your family. And there's nothing wrong with that if you really want to know about your family. God help you if you do, okay? But anyway, there's our...
Just go to Thanksgiving dinner.
Yeah, if you want to know, find all the, yeah. All right. So here, all I've done is they've represented the same information in a different way. When they shade in, that means you have affected, you have whatever the trait that you're interested in assessing. So these guys are heterozygotes, that's why it's half shaded. And there's their kid's outcome. Again, this doesn't, here's 3 children, so in this scenario, they had 3 children. In this scenario, this is hypothetical. This is what could happen. In a pedigree, it's what did happen. So in this situation, Joe and Mary, as carriers, because that's what we call heterozygotes, they carry the recessive allele, right? Those carriers had 3 kids. 1 was normal, well actually 2 were normal, and 1 had the disease. 1 was a homozygous, 1 was a heterozygote, and 1 was a homozygous recessive. All right? Now, I'm not trying to make this sterile. This is not fun. I mean, by the way, genetics is not a fun thing when we talk about diseases, but it's a very important exercise to do. All right? So all this just reminds you again, autosomal and recessive, both parents have to have a recessive allele. I often say this to students, it's just logic or common sense. If she was a heterozygote and he was a homozygous dominant, they couldn't have any kids with cystic fibrosis, could they? Because to have a child with cystic fibrosis, I have to have 2 copies, and each parent must give 1. Neither parent gets to give everything, right? They give what they have. Half of what they have, I should say. All right? So one more time, we learned this in meiosis. The sperm or egg will have 1 and only 1 of the alleles, and that's why I make Punnett squares this way. Yeah.
So if only one of them was a carrier, can you give birth to a carrier still?
Yeah.
You just
No,
couldn't have a
yeah.
kid
You just
that
could not have a
had
child
cystic-
with them. Yeah,
because
oh,
of-
absolutely. Absolutely.
Well, no, you wouldn't all be, no, because you don't have 25% being a carrier, wouldn't you?
Yeah, well, here, if you did again, you just replace that with a plus, and so all these pluses would go away. So 50% be carriers and 50% would be normal.
Oh, okay.
Yeah. That's how, I mean, that's just the basis of simple genetics, and that's all we really want you guys to grasp. And as I said, Punnett squares and pedigrees, these are just tools for organizing the information. They're just an end to me. You don't have to put anything in a Punnett square. You can do things in your head too. I mean, as you get used to doing genetics. It's just when you first are exposed to genetics, often students want to do Punnett squares because it's a way of organizing. And that's great. Whatever gets you the answer. Okay. All right. Now, autosomal dominant. So that was autosomal recessive inheritance mode. All right. Now, autosomal dominant, because we do see these as well, right, in the population. And this means that, again, autosomal, that the allele is somewhere on chromosomes 1 through 22. That's what the autosomal means. And dominant means that all I need is 1 copy of the abnormal allele and I have the disease. Because that abnormal allele dominates. Right? Just, again, when we talk about dominance recessiveness, we have to determine that when the alleles or genes get together. So, here in this scenario, things look a little different, right? Still Punnett squares, right? Still pedigrees to figure this out. It's just, I have to tell you, if I wanted you to set up problems like this, I'd have to tell you, is this autosomal recessive or is this autosomal dominant? I'd need to tell you that, because you need, that's vital to you being able to figure out what it means. So here, again, Huntington's is a case of, it's an autosomal dominantly inherited. So we have this unaffected parent, homozygous, right, recessive. The wild type is recessive in this case, heterozygous. And they have the disease because this one masks and dominates that one. All right, same thing, they go through meiosis, same idea. I'm going to separate their alleles, it's just chance. And here I've got the situation that you were asking about, Sean, right, on the previous one. I've got half the kids, they're homozygous recessive, phenotype normal. And half of them are heterozygotes, and they have Huntington's because of the influence of the two alleles when they get together.
Nick, would that go up to 75% if it was HD plus on each one?
In this case, and you're basically doing the same thing. Like I said, you'd see the HD here. If I made that one a plus, right? HD here, right? Yeah, so you have, well, you'd still have, no, you still have this one. You still have that one, so it'd be 75. Yeah. Because if that's an HD and that's an HD, then yeah, that one would, that's the one that would change. I mean, for phenotype.
Yeah.
And this one would still be normal. Right? So it's almost the opposite, I mean, basically the opposite of the autosomal recessives. All right. Now, there are exceptions, clearly a lot of exceptions to this, and I don't want to spend a whole lot of time because we just don't have a lot of time. So I took out a few slides that you guys probably still have access to. And I apologize, it's in the interest of trying to inform y'all. And so here we're looking at incomplete dominance and codominance. So there's different relationships between alleles, is what I'm saying. Okay? And again, how do we figure it out? We watched what happened when these were inherited in people. So when we say something is codominant, and this is your, right, this is basically the idea of blood type, right? AB is a codominant trait. You express both the A antigen, which is a protein, and the B antigen if you inherit those alleles, right? So blood type is an example of a codominant trait. Incomplete dominance means that when the two alleles are together, they don't both get to be expressed in their entirety, but they both contribute something to the phenotype. I know it's kind of gets a little confusing until you start to look at examples. All right, one of the classic ways we did a lot of work again in plants and fruit flies and mice because they have very high generation times, and we don't have any ethical problems with breeding them. We have ethical problems with humans, right? We do have standards in science, right? So we realized that in snapdragons, this was a classic example in snapdragons, when you got this red flowers and white flowers together, they'd always produce pink. So they wouldn't, neither of those alleles would dominate in the child. We'd get this partial, and that's what we call incomplete dominance when they're together, right? Codominant, again, means that both get to be fully expressed, all right? So this is, again, your ABO blood type example of this.
So you're saying the skin tone is an incomplete dominance trait?
Well, actually, skin tone is like at least three sets of alleles in the environment, so it doesn't fall into this, okay? But this is actually an example of familial hypercholesterolemia, right, FH.
So is it just pathogens then?
No, it can be any trait. We're just talking about a mode of inheritance. And when we say a mode of inheritance, we want to know how do these alleles interact when they're together. Okay, that's really what we're after. Okay, not trying to make a big deal over any of this. It's just reminding you that a lot of what we see in the world is not simple Mendel. Mendel was real important, but it's not simple. All right. In this example, when we're looking at familial hypercholesterolemia, the heterozygotes here are in between the two. Neither one, it's not a codominant, it's somewhere in between, like those pink flowers with red and white. This is quite a bit more serious, obviously, than petal color, but that's the idea. That the heterozygotes were somewhere in between a homozygous, right, with the abnormal and a homozygous normal. All right. Other factors, and I also, like I said, I think I left the slides in, but you can X through them if you so want. I'm not going to ask you a question about pleiotropy, expressivity, or penetrance. All right? They really are just degrees of influence of alleles on each other. Right? We use this term pleiotropy for genotypes. Right? Pleiotropy is and it's genotype and phenotype really, because when we talk about pleiotropic traits, Marfan's is one of those. Marfan's is actually affects one gene for elastin in the body. But it manifests as a number of different phenotypes with elongated and joints that are very, very loose and elongated limbs and heart problems because of all the elastin throughout the body. So pleiotropic effects are usually there's a single or it's a gene to a trait, and it's these genes affecting this trait, but right, it manifests with a whole lot of phenotypes. We also then have other examples like your book talks about deafness. Deafness is the phenotype is deafness, but there's lots of different alleles that can lead to that. So we really have to dissect away and try to figure out what genes are responsible for what and how do they interact together. All right? So I do add that environment influences genes. This is why it's not easy to say to somebody, well, you have BRCA1, you're going to get breast cancer, you might as well have a mastectomy right now. We can't do that because we don't know what environmental factors will either turn on or off this abnormal gene and its expression, because it's the expression of the gene that matters. All right, multifactorial traits, that's us, right? Most of disease falls into this. This is why, if you ever wondered why clinical healthcare isn't quite where it needs to be, well, part of it's because we have to figure out all the genes leading to heart disease, right? When I left UT Southwestern back in 2002, we were part of the Reynolds Project. So it was heavily funded. It was with either Duke or Chapel Hill and then California Institution, us, and one other United States institution. And our charge was basically to define, right, do gene screens of people who were susceptible to heart disease. And we were going to search these. I mean, we just did MRIs, we did gene screens, all kinds of different clinical endpoints as well as basic research endpoints. And I want to say the subject matter was in the thousands of people. And these are long-term, right, important studies for us. And our goal, of course, in the Reynolds Project was to try to figure out what was the genetic influence on heart disease, right? Because we still say that, or we did at least, that, 90% of hypertension is primary hypertension. In other words, that's the problem. We don't know, it's unknown etiology, isn't it, when you look it up? We don't know why. We suspect it's in the genes, but we don't know which ones. Right? So this is what we're after, what we're looking for. And
So
it's
did y'all
complex.
determine it?
What's that?
Did y'all determine that heart disease was...
I got to be honest with you, Sean, I
inherited?
didn't look at the results of the project to see. It didn't get published anywhere. I mean, it published through Google or any mainstream, so I'm guessing it probably wasn't as profound as we'd have liked, but...
Like Professor Myers will tell you very strongly that you cannot inherit heart disease.
I'm not going to comment.
I
Okay?
told my
There's a couple things
Disagree
going on
with
in Professor Myers' situation too, why he says that. But I'm just going to tell you that what I would safely say is we don't know for sure. Okay, but we're pretty sure most disease has. If we dig far enough down, we're going to find. Yeah. I mean, that doesn't discount lifestyle. I think that's the problem. Is a lot of times when people want to draw the lines and make it black and white, you can't do that. You can't make things black and white because there's a lot of factors that we don't even understand the interactions of all these that lead.
I was a big long ordeal with him about it, and he still disagrees.
It's okay. I mean, like I said, I did research for years. It's okay. You meet people where they are. Because that's how you begin to change mindsets.
So we're in church now.
Sometimes life is just like that, isn't it? All right. Okay, so multifactorial traits means I got lots of sets of genes that influence this trait, and the environment. Many factors. That's what it's right there in the name, isn't it? All right. So I say most, if not all, characteristics and disorders are considered inherited, and that reflects many sets of alleles and genes. And we got to figure them all out because they're not all on the same chromosome. That's why I said it was a complexity of Reynolds. You talk about individual variation. It's so hard to do these studies too because we've got to keep environments constant, and nobody can put forth that money, right? That'd be like creating the biodomes all over again, right? And sticking a whole lot of people in there and saying, okay, we're going to feed you this, and this, you're going to have this kind of air, right? You see what I mean? It's really difficult.
It's not exercise.
So we look for correlations, and then that's not a perfect world, but it's what we have. All right, now, I do make just a quick discrimination, just for terminology. Polygenic traits means I have many alleles, many genes. Many alleles, or sets of alleles, really, because it's always in the sets, because mom and dad, right, you get to give them at least one. Right? Sets of alleles, right? If I say it's multifactorial, I've thrown in the environment too, and the influence of the environment. So just to get the vocabulary straight, not going to ask you a test question to tell me what's the difference between multifactorial and polygenic traits. Not going to ask that, all right? So here's our rule of thumb, that if you're a multifactorial trait, you must be polygenic. But all polygenic traits do not have to be multifactorial, because there are traits that the environment doesn't influence. And here's one I know, and this could change too, right? Because everything's subject to change as we figure out the complexity of these relationships. Because it's chemical relationships in a nutshell, isn't it? So, right now, we would consider something like eye color is polygenic, but not multifactorial, because I do not know of a circumstance yet where the environment changes the eye color of a child. Now, color subjective, so you may perceive a different color, but it's not changing the actual amount of melanin that they have. Right? We don't have these mood eyes. Remember the mood rings? Anybody dated by that? Yes. When are haven't they come back at one point? So, where were the pet rocks? What the hell with the whole electronic devices? Why don't those kids want those? All right. So, here again, traits molded by one or more sets of genes, and really sets of genes, right, and environment on them. Okay, here's a classic one again. If you're like going, wait, I just don't quite, I can't picture this yet. Okay, and I got to get to a couple other things before we run out of time. Height, right? People's height is clearly multifactorial because we don't have short and tall people. We have a lot of in between, okay? But on top of it, the environment is nutrition, right? So even if you have all the right genetic combinations, and of course in here we better have some growth hormone, right? Because that's going to be a big trigger for growth, right? Genes for growth hormone, but not exclusive. Right? So you can have all the genetic predisposition to be tall, but you may not end up tall, right? Because if you don't have the right environment, and here we're talking about the nutrition, because we needed that vitamin A and C and D, among other minerals, calcium, phosphorus, in order for you to attain that growth, right? In order for those genes to actually lead to the trait. You see what I'm saying? And we could go down this road as well with skin color. All right? This is looking at an old picture where they did the height continuum, right? Starting with these actually have numbers on them from, down here in the 4 feet or whatever the lowest, right? Apparently we have shifted a little bit because we don't have anybody at the 410 anymore. This is a limited population. I'm not going to speak to anyone. I've got a lovely, Viviana might be the 410, don't you think, Mark? Right? She'd be our outlier right here. Right? We just had, all right? But this shows you again, this is clearly not short or tall, it's a multifactorial. It's not just one set of alleles, it's many sets. And this is just looking again at, now here's the genotypes, and remembering that ultimately the phenotype, which are showing in color, depends on the interactions of these sets. Eye color actually has about 8 sets. Your book's a little off. There's about 8 sets of alleles we know that's attributed to eye color, all right? But, again, complicated. Now, matters of sex. We got to get through this, and we've got to get through chromosomal abnormalities. Got to do that. All right? So, in your somatic or non-sex cells, right, so that's everything but the sperm and eggs, you will always have your 23 pairs, and you'll always have an X and Y, right? If I want to know your gender at a crime scene, all I need is a cell from anywhere. And I know, because all your cells have all 23 pairs. The only exception to all 23 happens in meiosis, right? Where you get to give one set of your alleles to your child, right? So, but when I use the term somite, or somatic, I'm really talking about all cells except for your sex cells, your gametes, all right? So we refer to this as, right, X and a Y is a heterogametic. It actually does have a different term. I won't ask you that on the test, I promise you right now. You get a little bonus for being in class today, right? But heterogametic is actually the term we use for this. Or you have 2 X chromosomes and you're homogametic, right? You have the same ones. All eggs carry X. Mom only has X's, that's all she can give, right? Dad determines the gender. This is, of course, Dad has an X and a Y, he's heterogametic, and he'll give one or the other. And theoretically, half his sperm would have X chromosomes and half his sperm would have Y, right? Fair? We're thinking about genetic lottery. And sex is determined by Dad. All right? So this just goes through. Here's this idea. If I'm going to have a little girl, I've got to have an X from Dad and an X from Mom. Mom can only give X's. If I have a little boy, it's a Y from Dad. Dad determines gender, and Y tells you it's going to be a male. Okay? If there's a Y somewhere in there, you've got a male. It doesn't matter what they look like phenotypically, that's a male.
So what are these people who are born hermaphrodite born with? Are they XY still, or?
Well, they're in the biological term of hermaphrodite, they don't exist in humans. Okay, I'll just be honest there, because you'd have to have complete functioning sex of your of both reproductive systems.
Okay, well, somebody's born with both genitalia.
All right, so it's an
Exterior genitalia.
Okay, yeah, it's going to be some abnormality where they didn't get a complete or enough dosing. Because often I'll be honest with you, in gene situations, it's the dose, it's the amount, how many copies of the gene are made. Because we're leaving a lot of this out, right? We're just saying you have the gene or you don't have the gene, but you got to remember you got to transcribe that gene and then you got to translate it. So gene dosing is a big deal, and that's what I'm talking about there. So maybe you didn't make enough copies, right? And you didn't get enough of this hormone, right? Everything we talked about in this class comes from our genes so far, and what if you didn't make enough of that? It's a possibility that you didn't suppress one structure enough, and you didn't stimulate the other enough. That's what I'd hypothesize on. All right, so sex determination, this just shows you a scanning EM and a color coding of the X versus the Y. Yes, they have a big difference. X has about 1,500 chromosomes, Y has about 200. You won't have to know those numbers, all right? Just understand that that's why we use the heterogametic. They're not homologous in a male, all right? Maleness comes from the Y. There's a gene on here called the SRY gene, and it gets activated, remember, at the end of the embryonic period, going into the fetal stage, right? Because we don't know the gender of an embryo. We could if we took a cell, because the cells would have the right chromosomes in it, but we couldn't tell at the end of the embryonic. Starting into the fetal stage, then this genes on the Y chromosome start turning on. In other words, they get transcribed and translated, and they make chemical products that cause the testes to descend and the penis to enlarge. And then in a sonogram, we could tell that, right? So between 8 and 12 weeks, we can tell the gender because of activation of genes on this Y chromosome. If the child doesn't have a Y chromosome, that isn't going to happen, right? The gonads are going to stay up in the body cavity, the clitoris is going to stay small, right? The labium are not going to enlarge. We have a female. You guys with me? It's an act of chemistry. All right? So the X chromosome, as I said, all in italics, you don't have to know this has quite a few more genes than the Y chromosome. Most of these there isn't a copy, right? That's why we use this term heterogametic. So in other words, if there's an allele on the X chromosome and the Y doesn't have it, then the males, right, they just have one allele for a trait. If it's a sex trait, sex-linked trait. All right? So there are Y-linked as well. I'm not going to go into those. I'm just going to talk about the X-linked ones here. Hemophilia is an example of this. Right? X-linked, mom gives the X. God bless you. All the time. Whenever I talk about this, students really want to make dad responsible for this part. They're like, no, because dad doesn't give, right, an X to a son. He gives a Y. And if the alleles or gene is on the X, it comes from mom. These are inherited disorders, mom to son. Now in daughters, mom and dad both have to give. But in a son, it's mom. Right? I'm not trying to make women feel bad, but it's mom, all right? And we can still set it up like a Punnett square. Right? Each son has a 50% chance of receiving a recessive allele from the mother. If she has 2 recessive alleles, if she has a trait, if she's a hemophiliac, all her sons will be hemophiliacs, right? Period. If she's a homozygous dominant normal, no sons will have the disorder, right? It's mom to son. All right? Each son with 1 recessive allele has a disease because X and Y don't have the same alleles. So Y can't block anything on X. All right? Each daughter has a 50% chance from mom and a 50% from dad. And if she gets one copy, she's a carrier. So the same rules that we talked about for autosomal recessive are true for females. That didn't change. All right? One recessive allele does not develop because these are recessive. Again, X-linked recessive. And I would identify for that for you, or if I didn't, I would want you to ask me that. What is the mode of inheritance? All right? Now, last part, chromosomal disorders. All right, so X-linked, there's going to be a couple questions on the test about, X-linked. No Punnett squares are set up on the test, but for the X-linked traits. But you should know, pass from mother to sons, and sons, and basically why this occurs, since there's no equal, there's not two copies in a male. Male have X and Y, not XX. Now, chromosomal disorders, this is a deviation from the normal. So this is another vocabulary for you guys. Okay? So when we talk about your number, 46, and that's why I said you should know that number. When you walk out of a life science class, you should know 46 is a magic number. We also refer to this as euploid. The word euploid is sets of chromosomes. And 2 is a typical, right? 2 is typical for animals. And that's why we refer to us as diploid or diploid, if you've ever heard that. Because di means 2. You have one mom and one dad. You have 2 sets. And that number that comes from human, 23 and 23, is 46. So 46 is our euploid. EU means true or accurate number. If you're polyploid, you have additional sets, poly many. So you could have 69, right, or 92. And then you wouldn't be diploid, right? Diploid's 46, and that's 2 sets. And that can happen. You won't live, I guarantee you that, but it can happen. Plants are actually very good at polyploidy. Animals are not. This is a fatal problem. If this occurs, and it occurs because something goes wrong in meiosis, then the child won't develop. Right? The embryo, remember those statistics I said beating the odds? And I said half of those are from chromosomal abnormalities. And here we are. Okay? Having the wrong number means I'm aneuploid, because AN in front means without or none. Right? So if I have 45, 44, 43, 42, 47, 48, 49, I'm aneuploid, but I'm not polyploid, because the set is 23 for us. So we have 2 sets of 23, we're diploid, or our number 46 is our euploid number. So this is just vocabulary again. What does this mean? So if I say you're aneuploid, I mean you have the wrong number. If I say you're polyploid, you have additional sets of alleles.
Still the wrong number.
But still the wrong number, you got it. All right. All right. So here, I'll elaborate a little bit more here again. Polyploidy results from formation of a diploid rather than a haploid gametes. All right? So if for some reason the sperm and the eggs didn't complete meiosis, sperm or eggs, because it happened in either parent, you need to know that, it could happen in either parent. If that happened, then you're bringing in additional sets. If you didn't reduce your number, become haploid, right, we're going to become polyploid. Most embryos, fetus will die. Occasionally they will survive, but there's a lot of abnormalities. Aneuploid cells are missing a chromosome or have extra chromosomes. So not full sets, but wrong number. Now this is from something called non-disjunction. This term, non-disjunction. Junction means joining, right? So dis means coming apart. So this means you didn't, right? You didn't come apart correctly. When do you come apart? You come apart in anaphase 1 and anaphase 2. Right? Because meiosis had 2 stages. So non-disjunction can happen either one. If you have an additional chromosome, you're trisomy, 3 instead of 2, 2 is a magic number when you get done. Right? Because a trisomy would be when you form a zygote. So if you're missing a chromosome, monosomy. Okay, now to make sense of this, let's look at it from this picture, because this is real important to image this, and those the words on that page may not be very helpful. All right, now understand, you have 22 other pairs of chromosomes. But here we're looking at a problem with one of the pairs of chromosomes. It can happen in the other ones as well, but to keep the cartoon simple and to address what we know about trisomy and monosomy, it's 1. So you could call this is a pretty large one. Let's say this is chromosome 2. All right? Now, this is happening in Dad, so this is spermatogenesis, isn't it? Happening in his testes, in the seminiferous tubules. Trying to give you a little heads up for tests, right? Yeah, right? And he's producing, right, taking his diploid cell with 46, the spermatogonium, and he's making a haploid with 23, right? That's what had to happen for fertilization to occur. All right, well, what if during the first, right, anaphase 1, the 2 homologs didn't separate, right, and they're just color coding it. Let's say that red is the maternal and green is the paternal. Doesn't matter. Right? Chromosome 2. Each one should have gone into a separate cell. They didn't. They stayed together. Non-disjunction. These daughters go on into meiosis 2. I don't have any copies of chromosome 2 to start with, so I have none when I get done, right? I had 4 copies, and 4 divided into 2 means each of my sperm has 2 copies of chromosome 2, and they were only supposed to have 1. All the sperm in this case is abnormal, isn't it? All of it. All right? I have sex with a euploid, normal, so 1 copy of chromosome 2, egg, and so the zygote here, this is a monosomy, has only 1 copy of chromosome 2, because mono's 1. That one's monosomic. These 2 are trisomic. You get how that works? It's math. It's just, there's 2 in there, mom brings 1 copy, I got 3 copies now of this. I'm not polyploid, but I'm aneuploid, right? And I'm specifically trisomy here and monosomy here. All right, this problem can occur in meiosis 2. So, again, homologs together in my spermatogonia. They separate like they should have in meiosis 1. This one goes through meiosis 2, and it turns out normal. It splits the centromere, each gets 1 copy of chromosome 2, 1 copy. This one does not split, so 2 of them go into 1 of them, the other one gets none. Again, normal fertilization occurs, so here, each of those eggs should have 1 and only 1 copy of chromosome 2. So I got 2 from Dad, I got 1 from Mom, I got 3. I'm trisomic. None from Dad, 1 from Mom, monosomic. 1 from Dad, 1 from Mom, euploid, diploid, right? Euploid, diploid. All right, does that make sense to you guys? I mean, because it's real more, like I said, it's much easier if you look at the picture-wise than if you go back to something like that and go, what? That's what happens. All right, now, here's the reality. And you go back to the karyotype to appreciate this, right? That trisomy, so one additional chromosome, right? Because 2 is a magic number. If I have 3, we're not. That's not good. 1 additional chromosome 13, 1 additional chromosome 18, and 1 additional chromosome 21. Because children born with these chromosomal abnormalities share similar traits and appearances, we call them syndromes. That's what a syndrome means. You have similarities even though you're genetically not from the same parents. You're genetically or even related. I say here, incidence at birth, maybe 1 in every 12,000, 1 in every, 8,000, 1 in about every 800. What does this tell me as a researcher? It tells me that when I add additional copies of these chromosomes, the outlook is pretty fatal. Because these probably happen as often as this does. It's just that when they happen, they often stop the development. Again, what we talked about in beating the odds. Chromosome 21 is a small chromosome, isn't it? Because the chromosomes are numbered from largest to smallest. And 21 actually is the smallest. We made a mistake between 21 and 22. It survives. These kids survive because, right, not as much is affected in their development. But they are a syndrome because even though they're genetically unrelated, they have a lot of similarities when you look at them. You can spot a person with trisomy 21 fairly easily in the crowd, can't you? Right? And this then talks about, and I'm not going to ask you to know these numbers clearly, all right? This is beyond what I need. But it illustrates, again, why. And we don't even see other examples. I mean, sometimes there's some examples of 15, and there's some examples of 22. But all of them have the same similar fate. They all die very early on. Okay? So this is a programming problem that is fatal. All right? Less devastating are the sex chromosomes. And why? Well, because the information on the sex chromosomes isn't so important to form you. Right? And all that development that went on in chapter 23, that was all dependent on genes turning on and off at precise times. Genes carried on these chromosomes. So if I have extra copies or missing a copy in the sex chromosomes, well, the genes on there are not as vital to my existence, are they? They don't define like where my heart ends up and the type of fibers in my heart. You see what I'm saying? So we see a lot more examples of aneuploidy, right, because it's still aneuploidy, here. And so I'm going to have a test question or two from recollection on just knowing whether they're monosomy or trisomy. So here's the reality. Turner's is the only monosomy that we know. There are no autosomal monosomies that we're aware of. The last slide showed you trisomies of the autosomes, right? Because 15, 18, and 21 are autosomes. There are no known autosomal monosomies. We're missing too much information for development to proceed. Okay? But in Turner's, this is a female because she's called, it's X. She's an X. As I say to my students, we have no yo, right? There's no Y and Y only. We often report that Turner's is XO. But there's just one copy of the X. And there is no examples of males that are monosomal. There's something on that X chromosome that has to be there still. Right? Klinefelter's, Jacobs, and Poly-X, clearly in that name, are all trisomies. All right? This is considered XXY. Please, if you haven't written that down, that's what it's, trisomy XXY. So there's two X's and a Y. So this is a male, isn't it? Y is male. Jacobs is XYY. This is a male as well. Poly-X, or triplo X, is XXX. All three are trisomies. They'd all have 47 chromosomes, right? She'd have 45. They'd all be aneuploid. And they would all be the result of non-disjunction in meiosis 1 or 2. They'd all be the result of non-disjunction in meiosis 1 or 2 in either mom or dad, except for Jacobs. Jacobs, we know, is dad's. Because dad's the only one that gives Y's, and he's only ever supposed to give one, and they have 2. Otherwise, we wouldn't know which parent was that the problem occurred. Not that it wouldn't really matter in the end, right? Because it's there. Now, I'll also let you know that you couldn't tell in a population readily, especially before puberty. You couldn't tell these people. They develop fairly normally. Turner's, because they're missing that, they're monosomy, and the only monosomy, they'll never go through puberty. She has no fertility. She'll never develop breasts or large. She'll just look like a prepubescent girl through her life. All right? These guys will all develop normally. This one will have compromised fertility. These two will not. And for the most part, they may never know that they carry extra chromosomes. It may never become apparent to them. In some cases it is, in some it doesn't. Okay? So the last thoughts here, because I've got read these two slides, just I know I have a test question on this. Might have two, and I appreciate you guys giving me a little extra time. It kind of took us a little time to get started here today. So prenatal tests. Now you're back to the question that Sean asked about. We don't do typical gene screens on kids. It's not that it can't be done. It's just it might be being done. I'm not aware of it. Even though it's a mainstream. These are mainstream, right? This is chorionic villi sampling, and this is amniocentesis. And we offer this to women who are over the age of 35 and 38. It's offered to any woman who wants it. It's just insurance covers it if you're an older mom. All right, I'm just going to tell you that's the way it works. You can hold your judgment, your fairness, whatever else to yourself, but that's the way it works. Because the villi is the chorion, that's the baby's cells, right? If I sample, if I pull through a syringe some of these cells, I can grow them in a culture. The cells, this can be done a little earlier than this. That's why it shows you this way. This has, last time, and this just depends on what the sources you look at, but last time this has about a 1 to 2% incidence of spontaneous abortion when it occurs, because it does disturb the uterus. So you better want to know that information, because you might lose a child. That's what it tells you, okay? Amniocentesis has a risk factor too. It's just a little bit lower. All right, because if you stick a needle in there, disturb that, that should seal back up. But you are disturbing the uterine environment, and you can cause the abortion of the child. Right? So again, when I'm saying spontaneous means it just the uterine contractions begin and you lose a child. You can't stop the delivery. Okay, I don't mean that in the sense of somebody choosing. Okay. So in amniocentesis, they draw some amniotic fluid. In the fluid are cells that came off that were shed by the growing baby. You culture them. In either circumstance, you have to culture the cells. So after you take them, you have to grow them in culture for a couple of weeks. And you get them to go into mitosis a few times. They'll have to go into mitosis again because you got to get enough cell numbers. And you do a karyotype. And so from these two procedures, you could, right, or would, if they got a good sample. And if they didn't, they'll go back and they'll try to get it again. All right. We're going to be able to tell whether or not there's a chromosomal abnormality. I can't tell you about a genetic problem. I can tell you whether or not there's too many or too few chromosomes, or there's a break in the chromosomes. Major, gross problems would be evident, but not singular genes, right? These are not, and these are pretty common practice. Now, if you've been pregnant here, you may not have had these done. I mean, I didn't elect to have them done when I got pregnant at 38. I said, no, there's nothing I could do about it. So, I mean, for the pregnancy, I had no intention of terminating the pregnancy, so I really didn't want to know, right? There was something I'd have to deal with it in time, and it wasn't important to me to know it, because I couldn't change it, right? And I also knew that there was a chance of abortion from the, this is spontaneous loss of the child from the procedure, so I said, no, don't need to know. But typically, most women have this done, where they have a blood draw done and they do a fetal screen for certain fetal proteins. Because some of the cells do actually cross into your blood from the placenta, right? And this is one of the things we talk about in the autoimmune diseases. That sometimes those cells, usually they're attacked and destroyed, but sometimes they persist and can lead to autoimmune diseases like scleroderma later in life, right? But typically, during the pregnancy, there'll be some cells in your blood. All right, not a high level, but in there. And we can also use those cells, not for amniocentesis. We don't do them for chromosomal abnormalities. We're really looking for the proteins. This is a fetal protein screen that we do. And there's about 4, at least there was when I was pregnant. They might have increased this by now, right? But there are about 4 proteins that we could do. Now, if you get the cells, you can do chromosomes. But that is not done in every pregnancy. But the serum proteins is done. So here, when we look at this maternal serum markers, right, amniocentesis, chorionic villi sampling, right, not the drugstore, right, and fetal cell sorting and ultrasound. The reality is that amniocentesis and CVS, this I could look for karyotypes. If I did fetal cell sorting, I could do a karyotype. If I get fetal cells, I can grow them. But most women do a blood draw, which is different than the fetal cell sorting. This is a more costly and expensive procedure. Most women go in and they get a maternal screen. They get a blood draw, and that's why I was trying to bring that back on this. They get a blood draw, and then we run typically about 4 proteins that we're looking for to see if they're elevated, increased, or decreased in her blood. That would indicate that there might be a problem. There is some chemical signature to trisomy 21 that sometimes shows up in the blood. There are some signatures to trisomy that show up on a sonogram too, by the way. So a trained sonographer can actually may be able to pick up these features on children that have trisomy 21. All right. Thank you so much. And I apologize again. Last time we had to have but I'm sorry I had to go over a little bit.